Hi, everybody. I am Kay from the Royal Society of Chemistry, and we will start very shortly. I'm just going to wait for the number of people to stabilise. Okay, we will begin. Hi, everyone, and welcome to today's desktop seminar by the Royal Society of Chemistry. My name is Kay Burrows, and I am the Deputy Editor for the RSC Interfaces portfolio. The webinar series showcases leading research in topics across the chemical sciences, and today we'll hear two amazing speakers discuss their work on active matter. So we have Professor Kathleen Stebby and Professor Sujit Datta joining us live. This webinar is scheduled for 1 hour, and each presenter will give a talk followed by an interactive Q&A session. At any time through the webinar, please submit questions through Zoom's Q&A function, and they will be asked after each presentation. First, there is a little bit of housekeeping. So we must note that the webinar is being recorded and the recording will be made available afterwards. Because of this, everybody will be muted and I will relay the questions during the Q&A session. We want everybody to be able to enjoy and contribute towards this webinar, so we ask all participants to be respectful of one another and to follow the agreement on this slide. Before we start, I'd like to highlight that the RSC has a variety of journals relating to the different kinds of active matter that you'll see in today's webinar. Specifically, I'd like to shout out Soft Matter as our home for research on colloids, polymers, and other materials and their behaviour. We've been going for 20 years and we have a very exciting themed collection at the moment celebrating our 20th anniversary. And we have published about 80 papers and over half of those are open access. So we really hope that you'll consider Soft Matter for your next submission. Without further ado, I would like to introduce our first speaker, Professor Kate Stebby. She's the Richard and Elizabeth Goodwin Professor of Engineering and Applied Science in the School of Engineering and Applied Science at the University of Pennsylvania. She works on directed assembly in soft matter and at fluid interfaces, and she is a member of the Soft Matter Advisory Board. I will now hand over to Professor Stebby. Thank you.
Thank you. I'm going to share my slides. Okay, let's try that again. And let me know if they project properly. Can you see it?
Yes, it's
Perfect.
now gone into presentation mode. That's perfect.
Okay, thank you all. And I just want to thank the Royal Society and Kay for having invited me, and Jim and Kay both for their care in setting up this seminar and tending to us at this late hour in the UK. I want to talk today about interfacial flows that are generated around colloids at fluid interfaces. And I want to talk about two themes. I want to talk about active colloids and biomixing at interfaces, and Brownian colloids and what we can learn about surface rheology by looking at the flow fields generated around them. So I'll start with biomixing because I've set up my title slide to motivate some of what we're thinking about. We're used to thinking about how to modify fluid interfaces using some additives, where we might talk about something like a surfactant head group and a surfactant tail and ask ourselves, how should I tailor the surfactant to get some mechanics or structural behaviors? Here, instead, we're looking at a bacterium that's located at the interface between two fluids. And the bacterium has some cell body, which will situate in some way in the fluid interface, and some means of propulsion, for example, a flagellum that might be located in one of the fluids. And we can ask ourselves, how might I want to design a trapping state to locate an an active colloid's body and its propulsion mechanism in order to generate mixing in the interface. And so we're looking at the bacterium Pseudomonas aeruginosa trapped in hexadecane water interfaces. And you're seeing a top-down view of these creepy crawlies swimming around on the fluid interface. And so one question is, how do they swim and what are their swimming behaviors? And then a more subtle question that will allow us to start to think more deeply about the problem is, what flow field is generated around them as they swim? And what can we learn about the system by studying this flow field? The work, I want to acknowledge my funding agencies. The active colloids work was funded by NSF's seabed grant, noted here. The rheology work was funded in part by that NSF grant and in part by a DOE BES grant on some functional molecules, which I'll talk about at the end of the talk. So with no further ado, let's think about why are we interested in the question of biomixing? And there was tremendous activity in this space generated by observations and analyses like the one that was presented in this now more than a decade old letter to Nature, which was about how living things in the ocean as they swim can displace mass to generate mixing that can rival the winds and tides. So with this exciting idea, people who like to think about active matter got involved, and there was beautiful work that was done also by, for example, thinking about what happens if a colloidal scale swimming agent is present in the fluid. And so I think it's a very interesting space, and we were asking ourselves when we became aware of the domain, how would interfaces be special? What might be different for biomixing at a fluid interface? So if I have a swimmer at a fluid interface, one question is, does its contact line become pinned, as is common for everyday colloids like polystyrene beads when they get trapped at a fluid interface? They make a hole in the interface and they get stuck there. There's thermal trapping. Their energies of the capillary trapping are so much larger than KT that they can't escape. And their contact lines become pinned at locations far from their equilibrium. So whatever height to which a colloid adsorbs on a fluid interface, it can stay there for some prolonged period of time. So you can get polydispersed behaviors from a single colloid. Does that happen also with a bacterium? If they are trapped at the fluid interface, does the high surface tension of the fluid interface constrain their motion? So do they have pin contact lines and constrained motion vertical with respect to the interface and in the interfacial plane? So how do they move? What flow fields do they make? And how is it different from the bulk? So I've mentioned that fluid interfaces are special domains. They're a fluid space, they're a space with very high interfacial tension, and they tend to become incompressible, a 2D incompressible sheet in the presence of scant surfactants. Swimmers on an interface can be stuck on the interface and yet remain mobile if that interface is fluid. And their motion can be constrained, as I mentioned, because of surface tension and contact line pinning. And if we do understand mixing at interfaces, it might be particularly interesting, especially for things like chemical engineering. There are tons of interfacial properties and processes that occur in chemical engineering. For example, simultaneous reaction and separation is a way to run chemical processes that are much greener than relying on reaction and subsequent separation. So we study the Pseudomonas aeruginosa. It can spin its flagellum in one direction, expel fluid, and move forward in a pusher mode. It can spin its flagellum in the opposite direction and move backwards in what's called the puller mode. And so if we look at movies like the one I opened this talk with, we can classify the different trajectories, the paths swum by the bacteria, in 4 different categories. There are those that are inert at the interface. Perhaps they're trapped so that their motor can't get access to the fuel that they need to be driven in the aqueous phase. They move in diffusive random walks. So they're just like passive colloids in terms of their behavior. There are others that spin furiously like a ballerina on point, who's so on point that she can't translate anywhere, but she just hops about diffusively as she spins. And we think of these as vertically trapped bacteria whose bodies and flagella are spinning in opposite directions, but with no ability to create translation. There are visitors that are not adhered to the interface who come and go from proximate to the interfacial plane. But the gross preponderance of bacteria at the fluid interface swim in curly paths, and each population remains stable over times of up to hours. And so we see that once a curly, always a curly, unless you get so exhausted that you become a diffusive random walker. This suggests that our bacteria, these diverse swimming behaviors, are taking place because the bacteria, once attached to the interface, maintain their interfacial attachment and their interfacial pose. And so pirouettes spin furiously, and they have roughly circular spots that their cell bodies make on the interface, whereas curlies have more elongated spots and are able to move more effectively. I focus now on curlies for the rest of the talk. So here's a curly trajectory. Here's a bacterium who swims with a curly trajectory. We can see that he goes in one sense, and then he switches the sense of his flagellum and goes in the opposite sense. And we can do analysis on these trajectories. We see that the bacterium moves ballistically and then becomes caged as it starts to swim in circles, because swimming in circles is a lousy way to explore space. And then afterwards, because of the diffusion of its circle's center of mass, it becomes diffusive over long lag times. And we can identify from whether it's going in one direction versus the other, and from its curvature behavior, whether it's swimming in a pusher versus a puller mode. And so we're going to focus on pusher modes and start to think about understanding the flow that's generated by bacteria, which are expelling fluid behind them and propelling their bodies forward by consuming fuel, just model active colloids. And so what we want to do is understand the flow field generated by these pushers. Do they move like classic Stokeslet flows in the bulk, where they would expel fluid behind them and they would experience a viscous drag on their body and make a very beautiful what's called stresslet flow field on the interface? Or do they have some more complex behavior? And even the idea of measuring the flow field generated by something like this becomes daunting. It's moving actively and diffusively. If we put colloidal particles around them, they will be moving diffusively because of Brownian motion, and picking out their signal would be very tricky. And so it's Mehdi Molay who came up with the idea about how to visualize this flow field. And he started using Brownian colloids. He said, let me look at a Brownian colloid that's undergone some kick. So here, let's call this the source colloid, and it's undergone a Brownian kick that moved it in this direction. And over here, at some position, I have a probe colloid, a tracer particle. And that tracer particle, by the rules of Stokes flow, is going to undergo some hydrodynamic displacement. So there's some correlation between this source's motion and this tracer's motion. But meanwhile, they're also both moving Brownianly. So in order to pick out the hydrodynamic interaction, the answer is to bin many such probes at this relative location with respect to the source. So Mehdi positions the source at the center of an origin and its displacement along the y-axis, and he bins many, many particles in a spatial location to pick up the hydrodynamic displacement that's generated because of our source colloid. And that binning is going to allow us to separate signal from noise. And the ability to just take tens of minutes of movies allows us to get really significant number of data points. So for example, 10 to the 6 in each bin will allow us to get nanometric resolution. So with that method, we can look at flow fields generated by Brownian colloids, or Jiayi Dang modified this method in order to look at flow fields that are generated around bacterial swimmers trapped in fluid interfaces. And there you see a movie of the colloidal tracers that she used around the swimmers to back out what was going on with the flow field that they generated. And So she used this correlated displacement velocimetry to image the displacement field, and that divided by the lag time is the velocity field generated by a swimmer in the interface. And we see it has some really interesting characteristics. It decays as 1 over r squared as expected, and it has a complex flow field, pulling in and being expelled from the swimmer, and these beautiful lobe-like structures that are generated around the swimmer. So we have the expected decay for a dipolar field, but not the expected shape. The expected shape would be this shape that I show here, some expulsion of fluid along the main axis and an influx along the waist. That's what we would expect in the bulk. That's what we'd expect if we had just surface tension characterizing our interface, but it's not what we see. So Nick Chisholm took on the task of trying to understand what happens if we have pinned contact lines and an incompressible fluid interface. And that concept is that scant surfactants on the interface can create such Marangoni stresses that the object moves, the active agent moves, it sees the interface as being incompressible. And so Nick was able to derive multiple dipolar modes that can be generated by swimmers in interfaces. One is an interfacial stresslet, which we show here, which has this beautiful 4-lobed butterfly shape. And that's what would be generated by in-plane expulsion of force by a pusher, drag on its body, and incompressibility generates the 4-lobe structure. There's another dipolar mode, which we call the B mode for a bulk effect. The flagellum is located under the interface. The Marangoni stresses that force the incompressibility are in the interface, and they give rise to this beautiful recirculating vortex flow. And if we superpose these two, we can recapitulate all the main features of the experiment. So this was Jai and Nick working together to find this nice parallel of theory and experiment. And now understanding the functional form for these dipolar modes allows us now to think about how could a swimmer generate mixing. So to summarize where we are right now, pushers at interfaces are trapped in diverse configurations. Their motion depends upon their trapped configurations. The interface is apparently incompressible, and trace surfactants are enough to do this, and the flow fields differ from the bulk. So our question now is, can we mix the fluids by hydrodynamic interactions? And we're going to focus right now on mixing in the interfacial plane. So the idea here is this idea of a Darwin drift, and I'm going to describe it using this very nice paper from JFM in the literature. It has a really nice graphic for bacteria swimming at 0 Reynolds number. I'll add that Nick Chisholm, who did the analysis for our swimmers, had looked at Darwin drift around swimmers at finite Reynolds number. So here we look at as well. So now let's think about this example. Imagine I took a spot, a vertical line in this fluid, and I just gave it marker particles. And then I asked myself, if a swimmer moves from far away along a linear path, how do those markers get displaced? And as that swimmer moves, the tracers are going to experience an outward pushing in front of the swimmer, a downward pushing, and then a backward pushing that's going to give us closed trajectories in the far field. Whereas in the near field, I can end up with net displacements. And this blue curve you see here is the path of net displacements, mixing open paths generated by the tracers, generating hydrodynamic mixing by the swimmer. We can do something similar for our swimmer moving along some vertical path with a tracer particle, and along finite paths, we can end up with openings or enhanced mixing. But what about circular paths? We see a preponderance of curly paths. And so we can also think about a swimmer moving in a circle. We can just think about a particle sitting in a flow field that rotates. And that rotation is going to have the particles see different pieces of that flow field as the rotation occurs. So Jai did the integrations to understand these paths. Here, the double blue loop is the displacement by our butterfly S mode. The red circle is the B mode, and the addition of the two is this funny little chin-like structure. This is reconstructed from her experiments, and we see that along closed circles in the far field, we're going to get closed paths. This is not great for mixing. If we're at finite separations, however, we can break the symmetry, generate open paths, and multiple circular swimming can generate helical displacements of our probes and really pronounced mixing. Active noise in the swimmer trajectories can also contribute to these open loops. And so we could look at now a tracer particle swimming with first few and then many swimmers, and that those swimmers have active diffusion. And we can see here with few swimmers, the tracers move in circular paths that are predicted by these swimmers, and few swimmer interactions let us have this caging or poor displacement and eventual enhanced diffusivity. As we increase the number of swimmers, we can eliminate this self-caging effect, and with strong active diffusion like that also generated by the swimmers, we can have first superdiffusive and diffusive displacements with a strong enhancement of the hydrodynamic diffusion of the tracer particles themselves, saying that there is mixing in the interface. And this can be seen in our experiments, although our experiments are far more complex than the analysis, and can be also seen here in our measurements of the hydrodynamic enhanced diffusivity. And here's analysis that supports our findings. And so we can roughly double the diffusivity in the interface. Just a quick note, shifting gears. What can we learn from an interfacial Stokes slit? If we take a particle and we move it with an external force, then we're going to generate not these dipolar modes, but this Stokes slit or monopolar mode in the interface. And that mode is very sensitive to the surface viscosity. The surface viscosity is captured in what we call a Boussinesq length. And as the Boussinesq length increases, we're going to be having larger and larger surface viscosities. And the qualities of our flow field change from having really like here a wasp-like tight little waist to more hourglass shaped, and here just a thickening waist and longer range decay in the slower decay in the surface velocity. And we can make master graphs of the velocity, for example, along the y-axis in the y direction or along the x-axis in the y direction, and show that with very, very large surface viscosities, we would decay logarithmically. So if I look at my radial displacement from the origin normalized by the Boussinesq length, I can have ranges of logarithmic decay, as in Saffman-Taylor. And then as I get further and further, I can get steeper decays. And so looking at these features should allow us to quantify the Boussinesq length and therefore the surface viscosity. We've done that for some functional surfactants, which we call lanthanide binding tags, which we're using to bind selectively to rare earth elements for a green recovery scheme where we can absorb these surfactants with the captured ions on interfaces and capture them in bubbles and separate them by a green process. But we can look at, for example, LBTs that can, these special surfactants that can bind to our rare earth cations to make charged complexes or uncharged complexes. Charged complexes, here absent their cation, are viscous. Adding more cation become more highly viscous, and adding enough cation to cross-link the structures, an abrupt change in the features of the flow field, and we see the markers of an elastic interface. Uncharged complexes, we've become more and more and more viscous, very thick wastes here. It's not subtle at all. It's a nice way of measuring surface viscosities and understanding interfacial stresses. With that, I'll conclude, and I thank you for your attention.
Thank you so much for that talk. That was brilliant. That was very interesting. We have time for a couple of questions. So there is the opportunity to put questions in the Q&A section of the Zoom interface, but I will start with a question, if that's okay, which is, can you infer the behavior of the interfacial swimmers in puller mode by thinking of pullers as pushers in reverse? Oh, I think you're muted.
Yeah. So if we were looking, so the idea of a pusher versus a puller is that is the question of what's the sense of rotation of the flagellum. If the flagellum rotates in one direction, it expels fluid behind the bacterium and moves it forward. And if we rotate it in the opposite direction, it pulls fluid in and the bacterium moves backwards. And in the bulk, that's really all you do is you switch the sign on the force fields and then off you go. A pusher and a puller are just a switching of sign and everything is the same. And at fluid interfaces, it works out that's not true at all. So let me show you first a quick little snapshot of the the gods of PowerPoint are not loving me. The pushers are not pullers in reverse. They here, this is a nice shot. As a pusher, we had these nice stable curvatures. And then when we went to puller mode, we have this really strong change in curvature. And when we go back to pusher mode, we get this nice stable curvature again. And we can see this here. We have pushers moving in a direction. And then when we go puller, we get this spiraling in of our path. And then we change again, we're pusher with a much weaker curvature. And we think that what's happening is that there's an instability in an elastic hook adjacent to the cell body, and it's wreaking havoc with the puller. I don't know whether it's good for mixing or bad for mixing. This is our preliminary attempts at understanding the flow it generates. It's crazy town. And so, no, we can't think of them as one being just the other with a change of sign. They're just very different, and they might be massive opportunities for deeper understanding.
Oh, that's brilliant. Thank you. So I have a question here, which is, what are exciting open aspects of active colloids at interfaces?
Yeah, I think it's a really open world. It's a little bit the wild, wild west. I think I showed you, for example, that the pusher was so well behaved that we could apply all of these classical tools, and that the puller is just doing crazy town, and that to understand it in detail is not going to lend itself nicely to classical symmetric analyses. And so I think maybe one of the really interesting things is just to go full on, full bore discovery mode. Like, what is a good mixer? Should you put a bubbler in the interface? Should you put a Marangoni surfer in the interface? Is there some direct measurement we can make? Can we just go after this? It's a really nice applied functional materials question. What is the best thing to put there to make interesting things happen?
That's great, thank you. I think we've got time for one more quick question, which is, can the circularly moving bacteria be able to create a pressure drop along the interface such that there may be mixing of the fluids?
Yeah, I think it's a really good question. In the interface, the stress fields are really very special stress fields. So we have what's called a Marangoni. If we look at our tangential stress jump at the interface, there's a Marangoni stress, which is a gradient in surface tension that can be generated. And understanding that for concentration gradient driven stresses, we need to understand something about how does surface tension depend upon composition. And if that coupling between tension and composition is sufficiently weak, then our swimmers could create two-dimensional pressure gradients. But what happens, well, in a sense, they do create pressure gradients. This is a very strong coupling. It's a very strong coupling. And so we end up having a Marangoni number, which characterizes the magnitude of this gradient with respect to viscous stresses, becomes divergingly large, and our interface becomes an incompressible sheet. So in a sense, that's already happening. A two-dimensional incompressibility constraint is being enforced by surface pressure gradients that are regulating the surface concentration. So there's all kinds of interesting surface pressure gradients in this interface that are giving us these funny four-lobe structures that are probably implicated in the mixing. So I never looked at incompressible versus compressible interfaces. It's probably already in there.
Oh, that's brilliant. Thank you so much. We will need to stop there. So thank you again so much for your fantastic talk. And an email will go out after this event with some follow-up responses to the questions that we didn't have time to get to today. So let me share my screen again. So we will now transition to our next speaker, but I would just like to take a minute to highlight some of the RSE's multidisciplinary journals. So the flagship general chemistry journal, Chemical Science, is a Diamond open access journal, so it's always free to publish in and to read. We also have ChemCom that publishes urgent communications of outstanding significance across chemical sciences. There's ChemSoc Reviews, which publishes review articles covering impactful topics at the forefront of chemical sciences, and RSE Advances, which is our community-led open access general chemistry journal. So with that, it is my absolute pleasure to introduce the next speaker, Professor Sujit Datta. He is a professor of chemical engineering, bioengineering, and biophysics at Caltech. His work focuses on the transport processes of complex fluids, gels, and microbes in complex environments. He recently published in Soft Matter on the flow of worm-like micelle solutions in serpentine channels, and he is our 2024 Soft Matter lectureship winner. He is also joining the Soft Matter editorial board in September. Today, he will be telling us about bacterial growth in complex fluids. So I will hand over to you. Thank you very much.
Okay, thank you. Let me just share my screen. Okay, can you see my screen okay and hear me okay, Kay?
Yeah, you're great.
Okay,
Thank you.
excellent. Well, first of all, I just wanted to thank all of you for being here across multiple time zones and geographic locations. And I, of course, wanted to thank the Royal Society of Chemistry, Kay, and everyone for setting this up and for the wonderful invitation. I'm grateful to have this opportunity to share with all of you some of the work that we've been doing over the past few years in my group. And so Kate gave this gorgeous presentation showing you about how bacteria, as they actively swim and move, they generate all sorts of interesting flow fields and mixing behaviors. And that highlights how bacteria exemplify active matter by moving around, by swimming, and creating work in that way. Now, bacteria are also exciting as a model of active matter in a different way. Sometimes bacteria not only move and swim around, but they also grow, they proliferate. That's one of the other fundamental features of bacterial life and microbial life in general, is they consume nutrients and they use that to grow and divide and continue to proliferate. And so in today's talk, I'll focus on that second fundamental feature of bacteria as active systems. And I'll hopefully highlight to you how this simple feature of growth and proliferation can give rise to really fascinating effects, and how bacteria as model active systems give rise to really interesting new effects in active matter, and in turn, how ideas from active matter physics can help us understand the biology of these bacterial systems better. And so, the overarching theme of what we try to do in my group is, we're excited about these complex manifestations of bacterial life in the real world. For example, in soil, in the subsurface, in our bodies, where bacteria have incredibly important effects, both good and bad, right? But these are messy, opaque, complex systems to study. And so in the lab, we usually study bacteria in more idealized uniform settings, like in bulk liquid cultures, right? And that gives us incredibly important insights, but it's often hard to recapitulate the full complexities of the natural world. And so to try to bridge the gap between these two extremes, what we try to do in my group is we take inspiration from this lovely quote by the great American philosopher of life and Yankees baseball player, go Dodgers, Yogi Berra, who once said, "You can observe a lot just by watching." What we do is we develop experimental model systems where we can systematically control them and visualize what's going on, but we can incorporate various kinds of complexities inherent in natural environments, such as crowding, interactions with various chemical and mechanical heterogeneities, and so on and so forth. And so today I'll give you one example of how we've been using this philosophy to understand bacterial life in complex fluids. And really the motivation for this talk, the motivation for this work comes from this young lady here. This is my daughter, Aiko, who in this video is doing something that I find incredibly gross, but it's also incredibly adorable. She's blowing spit bubbles, right? Not only is this process cute, but it's also important for Aiko's health because mucus, spit, is an example of a polymeric fluid that's secreted by our bodies, right? The mucins are individual polymers. Here's an electron microscope image from the group of Katerina Ribik that shows how you have this molecular network, right, in mucus. And the properties, the mesh-like structure of this network helps to regulate how it functions as a biological barrier against inhaled pathogens. And so bacterial infections often arise in mucus in the body. And so we wanted to understand, how does microbial life look like in these complex polymeric fluids? Bacteria also inhabit polymeric fluids in many other contexts. For example, many of you might have heard of biofilms, in which the bacteria are secreting their own extracellular polymers. They often interact with environmental polysaccharides and organic matter, right? And so bacteria interact with extracellular polymers in many different settings. And so over the years, people have found that the way bacteria swim, right, their motility is dramatically altered when the bacteria live in these polymeric environments. But we asked, what about growth, the other fundamental aspect of bacterial life? How is that altered, if at all, by interactions with extracellular polymers, given that bacteria inhabit polymer-rich environments in many natural settings, right? And this is particularly important in biology because often even nominally motile bacteria lose their ability to swim and to move and in these environments, and so they settle down and just continue to consume nutrients and grow and divide. And so this aspect of proliferation introduces new features to the typical models of active matter that we think about. And actually, we wrote a little review article describing some of the new physics that arise when you introduce proliferation into active matter systems. And I encourage you to check out this review article if you're interested. But the question that we really wanted to answer is, how do interactions with polymers shape how bacterial colonies grow? And so this is a question that my PhD student, Sebastian Gonzalez-Lacorte, bravely took on. Sebastian is now Dr. Gonzalez-Lacorte. He just defended his PhD, and he was co-advised with my good friend and collaborator at Princeton, Ned Wingreen, who helped us with the theory and simulations that I'll present. And this work also has been in collaboration with the groups of Katerina Ribik, who's a biochemist at MIT, who specializes in the biochemistry of mucus, and Dr. Sridhar Mani, who is a medical doctor at Albert Einstein College of Medicine in New York, who also focuses on the gut microbiome. And this work, at least the first result from this project, was published earlier this year in this paper listed here, and I encourage you to check it out if you're interested in what I talk about. And so to start, Sebastian did the simplest possible experiment you can imagine. He took immotile E. coli. The cells are fluorescently labeled. They're expressing green fluorescent protein in their cytoplasms, so their cell bodies are always lit up. And he put them in a microfluidic channel, right? So just in bulk fluid with nutrients everywhere, and you just watch them under the microscope as the cells consume nutrients, grow, and divide, right? And in that case, when you don't have any polymers, you just have regular fluid everywhere with nutrients everywhere. Each cell grows, it divides, the cells separate from each other, they diffuse away, and that process repeats. And so you just get a random dispersion, right, of these disconnected cells. This is exactly what you expect. And then Sebastian did the exact same experiment, but now with one difference. He added mucus. This is colonic mucus at the same concentration that you encounter in the body. That's it. That's the only difference. And we're watching the cells as they consume nutrients grow and divide. But in this case, when you have the mucus added, right, Sebastian saw something that we did not expect at all. This is what he saw. So the cells are still consuming nutrients, they're growing, they're dividing, But you notice that instead of separating away from each other and forming this random dispersion, they remain stuck end to end, and they keep growing and dividing, right? There's local order, and they form these long, beautiful structures that we call cables. And these cables, I'll play this movie again, they elongate, eventually they buckle, they intertwine, they form this beautiful living gel. This is like an entangled network of spaghetti, but now the individual spaghetti, right, are made up of bacteria that have grown and divided and stuck in this elongated mimetic configuration. And so Sebastian did this experiment again, found this again. And then this, for example, shows you a 3D reconstructor of these beautiful cables that Sebastian saw. And so he said, well, maybe this is just a quirk of the specific mucus that we were using. So we tested a different type of mucus, saw the same effect. When you have enough of the mucus, the bacteria, instead of growing in these random dispersions, they grow in these beautiful cable-like structures. Well, you said, well, maybe this is just a unique feature of the biochemistry of mucus, right? It has interesting biochemical interactions. Well, so then Sebastian tested a synthetic polymer, polyethylene oxide, right? Which is a very commonly used inert, non-adsorbing, uncharged, linear, flexible polymer. He tested 3 different molecular weights for each PEO above a threshold concentration of the polymer. When we added enough of the polymer to the liquid, the bacteria grew in these cables. Well, he tested Phycol, which is a branched polymer. Same thing. Above a threshold concentration, the bacteria grew in these cables. Sebastian tested sodium carboxymethyl cellulose, which is a negatively charged polymer, to see if charge interactions play a role. They didn't. Above a threshold concentration of polymer, we saw cables again. Sebastian tested this not just with E. coli, but he also tested it with Pseudomonas aeruginosa, which Kate talked about, right? In motile, Pseudomonas aeruginosa is often implicated in lung infections. We saw the same thing. Above a threshold concentration of polymer, these bacteria grew into cable-like structures. He tested Vibrio cholera, which is a gut pathogen and a biofilm former. Same thing. When there was enough polymer in the surrounding fluid, right, these bacteria grew in these beautiful cable-like structures. And so these experiments told us that this cable growth, right, is a generic feature across different bacterial types, across different polymer types. Whenever these bacteria are growing in a sufficiently polymer-rich environment, they grow into cables. So why? Why are these cables forming, right? And so, well, one clue comes from just washing away the polymers. Sebastian grew cables, and then he washed away the polymers, and he found that when the polymers were removed, the cables just fell apart, right? So it's not like the cells are getting glued together. It's not like the cells can't separate from each other. They can, but somehow when there's polymers around, the cells are getting stuck in these elongated cables. So what's going on? What causes cable formation? Could it just be that the cells are getting stuck in the polymer network? No, we didn't find any correlation between the threshold polymer concentration for cable formation and C star, the critical overlap concentration when these polymers start to form a network. Could the viscosity, when you add more polymers, suppress the ability for these cells to diffuse away? Nope. We didn't find any correlation with the viscosity of the fluid. What about the osmotic pressure? As you add polymers, they're increasing the osmotic pressure of the fluid. Could that somehow be altering how the cells grow? No, it turns out that these osmotic pressures, because these polymers are so large, are often as low as tens of pascals. Cells change their growth. They respond to osmotic pressures, typically on the order of hundreds of kilopascals. So many orders of magnitude larger than the pressures that these polymers are generating. So what's going on? Well, many of you are soft matter physicists, and you all know that when you take particulates and you put them in a polymer solution, you get depletion interactions. And the idea behind this is really simple, and it's so beautiful. This is an attractive interaction that emerges from purely repulsive interactions. When you have 2 bacterial cells that are just randomly diffusing around in a polymer solution, right? They randomly come close enough together, and if the space between them is so small that the polymer chains can't fit into that space, then they're excluded from this region between the cells. That's called the excluded volume. And so as a result, the polymers are pushing on the cells everywhere else, right, due to their osmotic pressure, but they're excluded from in between the cells, so they push the cells together. That's an attractive interaction known as the depletion interaction, because the polymers are depleted from the space between the cells. And so you can write down an expression for what the strength of this interaction energy is. And it's given by the product of the osmotic pressure generated by these polymers and the volume of this excluded volume region. And now, thanks to decades of work by colloid scientists, soft matter scientists over the years, we now have ways to quantitatively predict and calculate these depletion interactions for cells of different shapes and sizes and polymers of different compositions and shapes and concentrations, right? And we now know how this depletion interaction depends on the properties of the polymers and the properties of the cells. And these are things that we can calculate, right? And we can calculate what the strength of this depletion interaction is. And as you might intuitively expect, it's strongest when these cells are side by side, because that's what maximizes this excluded volume. And so we thought, well, maybe the depletion interaction is playing a role somehow, but it was a little puzzling because the depletion interaction wants to keep these cells stacked side by side. But in these cables, they're growing end on. So what's going on? Well, we built a simple model to try to capture what we're seeing in the experiments, where of course you have growth and division. Each cell grows and divides after a doubling time TD. Of course, the cells are rigid objects, so they push on each other. We have the depletion interaction that we can quantitatively model thanks to theoretical work done earlier, right? And of course, these cells, they are random diffusive objects, so they can diffusively separate from each other. These are the four basic ingredients that we put into the model. Sebastian built an agent-based simulation that includes these four ingredients, and there are two control parameters that emerge from this picture. One compares the strength of the depletion interaction set by these polymers to thermal energy, KBT. The other control parameter compares timescales. It compares the diffusive time of these cells to rotate around and separate from each other compared to the timescale of growth and division, right? So those are our two control parameters. And when Sebastian coded this up, when you don't have any polymers, no depletion interaction, in this case, the cells just grow, they divide, they separate, they diffuse away, and you get this random dispersion exactly as we saw in the experiments without polymers. When you add polymers now, so you have the depletion interaction, lo and behold, you see something beautiful. These cells initially stack side by side, but then they keep growing end on, and you see cables form. This looks exactly like what we see in the experiments, but it still doesn't answer this question of why these cells aren't just stacking side by side. And I should point out that this is exactly what we see in the experiments, where the cells initially stack side by side as a doublet, and then somehow they get stuck in this interdigitated configuration and keep growing end on as cables. So there seems like there's something special about this 4-cell proto cable, as we call it. And indeed, it looks like they might be stuck in a metastable state. That looks like a metastable state. And we can test that using the simulations, right? When you have weak depletion interaction, so that's this blue curve, start out with these cells in this interdigitated configuration, then the depletion is so weak that the cells reorient and they eventually reach this side-by-side stacking that you expect from depletion. But when you crank up the strength of this attractive interaction, right, so that's the red curve, they remain stuck in this proto cable. So this really looks like a metastable state, and Sebastian quantified this further by mapping out the full energy landscape of these cells when you have these 4 cells in this configuration. And indeed, as you expect, this completely stacked side-by-side configuration is a global minimum. That's what the depletion potential prefers. But when you look at this energy landscape, what you see is that there is a local minimum, right? This interdigitated configuration is a local minimum. And so the cells, they get kinetically arrested in this local minimum before they can continue to explore this overall energy landscape. And so we can compute what the shape of that local minimum in the energy landscape is, and then just use very simple ideas, right? Thinking about this as an activated process activated by thermal energy, KT, we can compute how long will it take for the system to get stuck in this configuration before it can escape from this local metastable state and eventually reach this side-by-side configuration, right? And that, of course, depends on the properties of the cells, the properties of the polymers, and this is something that we can quantitatively predict. This escape time, how long it takes for the system to escape this local kinetic trap. And so now we have a very simple unifying principle. We have a simple biophysical prediction. If this escape time is longer than the doubling time, then the cells are stuck in this protocable and they just keep growing and dividing and cables form. But if the cells can escape this local kinetic trap, right? If that escape time is shorter than the doubling time, then we don't expect cables to form. And so we can quantify this prediction across many different cell types, polymer compositions, concentrations, and that results in this state diagram that I'm showing you on the left here. The x-axis shows you that comparison of energy scales. So adding more polymers, right, is going from left to right. That increases the depletion energy, right? Going from bottom to top describes the competition of timescales. So as the cells grow slower and slower, right, as the doubling time gets longer and longer, that's going down on this phase diagram. And this black line that you see here is that biophysical prediction for when that escape time equals the doubling time. And we expect that to the left of it, we shouldn't see any cables. To the right of it, that's when cables should arise. And that's exactly what you see by these color-coded points. The solid symbols are from the experiments. The open symbols are from the simulations. And in all cases, this very simple biophysical principle captures when or when we don't see cables. And so this gives us a way now to predict, right, across different polymer types, cell types, right, when we expect this really weird kind of cable formation to arise. And so that gives us a framework to now start to understand these kinds of structures that biologists have seen in many different contexts, but the explanation for why they arise has been unclear, right? But this is just the tip of the iceberg. We just studied one clonal population. So you can ask, what do these look for when you have mixed communities where you have different cell types inhabiting the same space? That's something we're working on. Of course, the picture that I showed you here neglects any specific biochemical interactions, which are certainly important. So how do those additional interactions change this picture? This is something we're also working on. And then, of course, the question is, in addition to the fundamental interest in active matter physics, what are the biological implications of this cable growth? Could it be that this is a defense mechanism? Hosts are secreting mucus to cause the cells to get stuck in this form where they can get easily cleared away or eaten by immune cells? Or could it be that it's somehow beneficial to the cells by promoting biofilm formation? These are questions that we're continuing to address. And I hope that you all also get inspired to try to address these questions building off of what we've done. Finally, I wanted to show one slide because Kate is the other speaker in this webinar, and I'm showing this just for Kate. In addition to what Kate presented with bacteria swimming at interfaces mixing up fluid, Kate's done a lot of beautiful work studying active matter in liquid crystals, where now the fluid itself has orientation, right? For example, the molecules of the fluid are elongated so they can align with the same orientation. That's known as a nematic liquid crystal. Many biological fluids that bacteria inhabit are liquid crystals. And so Sebastian asked, what if instead of polymers, which are just randomly distributed, what if you have a nematic liquid crystal? How do bacteria grow in a nematic liquid crystal? This is unpublished. We're working on this now, but what you'll see here is these cells, as they grow in this liquid crystal that's aligned left to right, they grow in these one-liners, right? Not as random dispersions. And then as they grow, really weird and beautiful things happen. I'll play this movie again. By the way, the gray, black, white, that shows you the orientation of the liquid crystal itself through cross polarizers. You see, as this chain grows, it eventually buckles locally. Those buckles feed back on the orientation of the liquid crystal, and you get all sorts of complex self-assembly behaviors. And we've been working with our colleagues Severi Spagnoli and Tom Chandler at the University of Wisconsin-Madison, and we've developed a theory that describes the competition between the growth stresses induced by the bacteria and the bending penalty associated with bending the liquid crystal around it that captures what we see in these experiments. And I hope to share more details of that in the future with you all. But with that, I hope I've convinced you all that you can indeed observe a lot just by watching. I told you about one story where we've applied this philosophy to studying bacterial systems. We've studied bacterial systems in many other contexts. We've also studied fluids and polymers and colloids and gels. And what we're trying to do really in our work is bridge the gap between studying these systems in idealized, simple environments in the lab and their messy manifestations in complex spaces in the real world. With that, these are the people who make all this work happen. Thank you for listening, and I'd love to hear your questions.
Thank you so much. That was a fantastic talk. We do have some questions. So the first one is, where do such polymer-induced cables appear in nature?
That's a great question, and that's something that we're still uncovering. Some of the actually, after we published this paper, we've been contacted by many microbiologists from across the globe who've shared with us observations that they made of similar cable-like structures, but they just didn't understand why. In some cases, cables seem to arise in biofilms. For example, in biofilms that form on teeth, like plaque, people have seen structures like that, primarily in biofilms, as well as in some kinds of biofilms that have grown in the lungs. But that's still an open question, and that's part of what we're trying to address now is now that we've discovered this weird form of growth in the lab, and now that we've seen that examples of it arise in nature, we're trying to use the quantitative framework that we've developed to predict when these cables arise, why they arise, to use that to understand their manifestations in the messy real world. So stay tuned, and I encourage you all also to look for cables in your own work if you study bacterial growth in complex fluids.
Oh, that's brilliant. Thank you. So the next question is, do the cable lengths follow an Ostwald ripening-like scaling, i.e. for the increase of length versus time, and have a power law-like dependence?
That's an interesting question. No, it doesn't look like that's the case. And but I think there could be examples where that does arise. In our case, because the cells are all in a nutrient replete environment, the cells are essentially living in chicken soup. They're in a buffet where they can just eat their hearts out. All the cells are growing exponentially. They're growing at their maximal rate. So these cables grow from these founder cells and they just keep growing and growing and growing exponentially. And so that's what we see in our experiments. And it looks like, each cable is its own kind of lineage, and they don't really exchange cells with other cables until they kind of crash into each other and then they coarsen and entangle. But there could be situations where that arises. And that's a really interesting question.
That's brilliant. Thank you. Okay, the next question is, what determines the direction of branching and linking of cables, and what happens to the cables when the basal bacteria chains divide? Can we thereby tune it to produce on-demand rheological properties?
Ooh, that's a great question. I describe these as living gels, and indeed they are living gels. And that's part of what we're interested in exploring. I'll just show you the 3D reconstruction of these because I think it's so beautiful. You can see that these things form these kind of entangled networks that are truly like gels, but these are weird gels. And so part of what we're studying, just to start to get at this question. But really, this is an opportunity for the entire field. There's only so many hours in the day. So we will happily share our protocols and so on with anyone who's interested in trying these experiments, right? What we're doing at the moment is we're just looking at the mechanical properties of individual cables. To what extent can we describe them, right, as semi-flexible elastic beams? And actually, it seems like if you blur your eyes and ignore the fact that they're made up of these individual bacteria, there's some aspects, some continuum mechanics that can be used to understand the mechanical properties of individual cables. But then the question arises, okay, when you have many different cables together entwined in this gel, right, in this living gel, what are the rheological properties of that gel? And that is still an open question. That is very much an open question. And that's something where we have ideas for ways to address that question. And I hope other people get interested in this as well and try to address that on their own. I do think that this idea of using these controlling cable formation to control the macroscopic rheology of these living gels is a really fascinating idea. And I think there's a lot to that. I think there's a lot of potential there. In terms of branching, I want to point out that these cables don't branch. We don't see branching, right? They start with these proto cables and they just grow along these lines. So they seem to form linear kind of filaments. But then, of course, as they grow, because of the compressive stresses induced by growth, they buckle. We have some a modeling of that process, but they also intertwine, they entangle, they meet each other, and they coarsen. And so other complex dynamics arise, but we've never seen branching from an individual cable because really they're getting stuck in these interdigitated proto cables. But overall, I think this question of rheology, what are the rheological properties of these living gels and how they can be tuned on demand, I think is a really fascinating direction to take this work.
Oh, brilliant. Thank you. I think there are some more questions, but I think in the interest of time, we will wrap up. So I will just have another slide. Hang on a minute. Okay, great. So that brings us to the end of this webinar. Thank you to everybody who's joined today. Thank you to Professor Stebbi and to Professor Datta for some fantastic talks. I've really enjoyed them. You can follow up. There are some email addresses on this slide, and we will send out an email after the event with some follow-up responses as well. And we hope to see everybody at future desktop webinars. The QR code on this slide has the information. We have a very quick call to action. If you're interested in helping us improve the publishing services at the RSC, then you can sign up to join the RSC Digital Research Lab. There's a QR code here as well. And that is everything. So I will bring this to a close. Thank you again, everybody, for attending. Thank you to our speakers. We hope you've had a great time and have a great rest of your day. Thank you.
Thanks, everyone. Take care.
Thanks. Bye.